Hello and good morning everyone. So we have discussed in the previous two lectures about the complement system. We have discussed about their pathway. We have discussed about the what are the biological effects that can be produced due to this complement pathway. They are the one of the protective mechanism that belongs to both innate and adaptive immune system. The classical pathway belongs to the adaptive immune system, whereas this mannose binding lectin pathway or lectin pathway and alternative pathway belongs to the innate immune system. They all have the responsibility of formation of this membrane attack complex that helps to lysis of the cell and kill the bacteria and then helps the immune system to eliminate the infection. They have other function like opsonization that also kills the organism. They clear out the immune complexes to prevent the type 3 hypersensitive reaction, and they have the chemical mediator inflammation to boom the inflammation. So these are the function which we have discussed about the complement pathways. Now you have to remember one more thing. This complement pathway activate only when there is required, there is a stimulus. But if it is activated without stimulus, then they will develop a lot of diseases. So that is also an important point of questions. So normally these complement proteins are present in the blood. They are in the inactivated form. They are not present in the activated form. They get activated only when there are the certain stimulus, like there is the antigen antibody, then C1 protein get activated, there is the mannose binding, then lectin get bind to that, then get activated this MBL pathway, and alternative pathway this C3b with the factor B and factor D get activated. So sometimes what will happen, due to the deficiency of the inhibitor, normally these all proteins are present in the inhibitory form. They are actually present in your blood, but they are inhibited by some of the inhibitor. They will only be activated when there will be the triggering factor, there will be the stimulating factor. So because of the stimulating factor, now they get activated and perform the reaction, the series of reaction, and clear the infection. But if without stimulus, in the resting stage, in the normal stage, if they get activated, then we will develop lots of diseases, and we will discuss about that. Okay, so we have known about this classical pathway and MBL pathway. In classical pathway, there was like in classical pathway it was the antigen antibody and this C1 QRS. Then in the MBL pathway, they doesn't require QRS, they only activate directly this, and in case of alternative pathway, even this portion is not required. They get activated directly and convert into the C3 convertase, and they finally after C3 convertase they will form the C5 convertase in both phase, and they cleavage the C5 and form this membrane attack complex. So this is the way. The main end product is same, but the problem is about their C3 convertase, which is different in classical and MBL pathway, but in case of the alternative pathway, it is different than that of this classical and MBL pathway. So biological effect, we have talked about this: opsonization, chemotaxis, anaphylatoxin, and cytolysis. They kill the organism, the membrane. This is known as the membrane attack complex. They attack the membrane and disrupt that membrane and cause the lysis of the cell, and bacteria or virus get killed. The opsonization, this is one of the way of enhancing the phagocytosis. Chemotaxis, anaphylatoxin, they bring out this phagocytic cell and cause the inflammation. So you can see over here there is the functions, like there is the membrane attack complex that cause cytolysis, and the bacterial cell get porous, the membrane is attacked and become pore, and then they kill the bacteria. They also play a role in opsonization, that is the C3b, and there is also inflammation that is taken place by the C3a and C5a, that is the anaphylatoxin. Okay, so opsonization, we know this is one of the way of increasing the killing of the pathogen. Complement has a receptor, one to the macrophages and another to the bacteria. They bind to the one side to the phagocytic cell and another side to the bacteria and kill the organism. So this is the one of the ruthless way. One side you tight the say bacteria, another side you bring the phagocytic cell and kill that organism. There can be also certain this C5a that brings they are chemotaxis, they brings out the phagocytic cell towards the inflammatory site. So we have discussed about this classical pathway, mannose binding MB, lectin pathway, and alternative pathway and their functions. as well. Now let's finalize. For final time we discuss about this function. This has the lysis, that is cause complement protein that cause the lysis of the cell. They can also help in opsonization. The activation of the inflammatory response bring all this chemotaxis, all the phagocytic cell towards it, and they also actually cause this clearance of the immune complexes. So these functions are actually very important. This complement protein is very important for us. Okay, now if you move forward, then we will have some of the deficiency. I have already told you complement proteins are actually in the inactivate form present inside our blood. They are not activated unless there is some stimulus. If somehow the inhibitory protein which are keeping them inhibited in the inhibited form in the normal resting stage is absent, then the complement path will be activated and we will develop many diseases. Like if there is a C1 inhibitor, that is the classical pathway which required C1 QRS, if that is inhibited, then this has irreversibly bind to inactivates the C1 QRS and QRNS as well as to the MSP in the lectin pathway. So both pathway will be activated if this C1 inhibitor is not there, and without any stimulus if they are getting inhibited. So see, this C1 inhibitor is actually inhibiting this QRNS to bind to it. If it is not there, then they will bind and the cascade of the series of reaction will take place and we will develop the disease. The disease is known as the hereditary angioedema. So this C1 inhibitor esterase has role in the bradykinin mechanism as well, and because of that they will develop the hereditary angioedema. So we have to understand this. Okay, so coming to the another point, there is a decay accelerator factor. We have talked about this classical and mannose binding lectin pathway. Then there is the alternate pathway. In alternate pathway, actually this delay accelerating factor DAF is present. This is also called CD55. So this protein is actually present to inhibit the formation of this C3b big B. So normally you know that C3b big B is the C3 convertase that is formed in the alternative pathway, and first C3b is there, okay, and then they get activated factor B and factor D, and then this big B small b comes. Normally this big B and small b cannot be bind to the C3b without any stimulus. So if there is no any microbial surfaces that is activating the alternative pathway, so there is not requirement of this activation. If you get activated, then there is no pathogen, but they will be triggered and they will kill your own cell or damage your own cell. So because of that, this DAF factor will prevent this formation of C3 convertase in the normal resting stage. But if this DAF factor is absent, then what will happen? The complement pathway will be activated. This DAF prevents the binding of factor B to the C3b. You can see C3b is here over. This DAF is preventing this big B and small b to bind over here in the normal resting stage. If by any means this DAF is absent, means this B gets chance automatically without any stimulus, they will bind to this and the complement pathway will be activated. The alternative complement pathway will be activated. So this DFE dislodges this C3b bound factor binding this big B and small b, and this is actually inhibited. When there is a microbial surfaces, an alternative pathway is activated, this DAF factor is removed and then they bind to each other and pathway is completed. But without the presence of stimulus, if they get activated, then they will develop disease. So what is preventing to get activated by itself? This is DAF protein. If we remove the DAF protein, or if there is a deficiency of DAF protein from the congenitally, then we will activate the alternative pathway will be activating in our body continuously, and we will develop the disease called the paroxysmal nocturnal hemiglobinuria. So what is happening? The alternate pathway is getting activated, they are damaging your own RBC, and you are developing this paroxysmal nocturnal hemoglobinuria. So you have to understand this concept very nicely. DAF protein normally prevent, normal resting stage, alternative pathway is not activated, but if there is a deficiency of DAF protein, then we'll develop this disease called paroxysmal nocturnal hemoglobinuria. What are the risks of having this PNS? They will be the blood clots, kidney damage, fatigue, lung problem. This all are the problems of the paroxysmal nocturnal hemoglobinuria, because our RBC are actually getting damaged. We will not have the enough oxygen and food, and then obviously there will be the fatigue, lung problem, kidney damages, and as well as the blood clot will occur. So there are other things as well, like propridine, C3b and stabilize C3 convertase. Factor 1 cleavage cell bounded or fluid phase C3b and C4b, inactivate C3 and C4b. Factor H regulate the alternative pathways. So these are the factors that have their functions. Amount of C5 convertase available, reduce the amount of C5 convertase available. So these are the normal function. But if this is absent or this is congenitally absent, then we will have developed the diseases. So that we have to understand. So now let's move to the regulation of complement system. C4 binding protein inhibits the action of C4b in a classical pathway, split C4 convertase. Complement receptor 1 cofactor for factor 1 together with the CD46 protectin, bitronectin. These all have the function that is normally regulating your complement pathway. If they are absent, then obviously we'll develop the disease. The diseases are like if there is a deficiency of the MAC complex, C5b, 6, 7, 8, 9, then we'll develop recurrent severe Neisseria meningitis or Neisseria gonorrhoeae infection. So we'll have Neisseria bacteremia if there is a deficiency of MAC complex or manus binding lectin. If there is a deficiency of C3, then we'll develop severe recurrent pyogenic sinus and respiratory tract infection. If there is a deficiency of C1 esterase, we have talked about this initially, it will develop angioedema, that is the increased capillary permeability and edema. If there is a DAF deficiency, the will develop increased complement mediated hemolysis, that is the paroxysmal nocturnal hemiglobinuria. There can be a transfusion mismatch, like activation of complement, generation of large amount of anaphylatoxin and MAC, that is the red cell hemolysis. There can be autoimmune diseases. If immune complement pathway is not intact, then their immune complex will not be cleared and they will go and dislodge into the small capillaries and will develop the autoimmune diseases. If there is a severe liver disease, what will happen? Complement protein will not form. So in the inactivated complement protein is also not there in the blood, and since despite the stimulus, if there is no protein, that will not be activated and we will have this recurrent pyogenic infection. I'll discuss about this transfusion mismatch in detail in a few minutes. There can be clinical aspects like factor 1 deficiency, which will low level of C3 in the plasma due to unregulated activation of the alternative pathway, that will cause the recurrent bacterial infection in the children. So this is the deficiency of the last component, that is the membrane attack complex and manus binding lectin. If it is deficient in your body, we will develop this Neisseria gonorrhoeae or Neisseria meningitis. This is Neisseria species recurrent infection that you have to understand. Then C3b, if there is a deficiency, will develop severe recurrent pyogenic or sinus respiratory tract infection. This is the complement protein deficiency. If there is, then we'll develop that. If there is a C1 esterase inhibitor, will develop angioedema. This is coming repeatedly. You can see the angioedemic picture. So this is the angioedema that is developed due to the absence of this C1 inhibitor esterase, because C1 esterase inhibitor, because they have the role in the bradykinin plasminogen and bradykinin mechanism as well. So there will the metabolism of bradykinin will be affected, and because of that, this C1 esterase will leads to the angioedema, because they will increase the capillary permeability and develop this angioedema. So decrease of this deficiency of DAF will allow the activation of your complement pathway, and because DAF is preventing in the normal resting stage to activate this complement, if this DAF is deficiency, this alternative pathway is activating continuously, and because of that will develop this paroxysmal nocturnal hemiglobinuria, and this will lead to that. These are the symptoms like dysphagia, pulmonary hypertension, chronic kidney disease, dyspnea, abdominal pain, and all other features: anemia, fatigue, impairment, erectile dysfunction, hemiglobinuria, thrombosis, and a lot of the complication of this is the due to these are the feature of your paroxysmal nocturnal hemiglobinuria, which was developed due to the activation of the continuous activation of the alternative complement pathway, and that was due to the deficiency of DAF. So transfusion mismatch reaction. Okay, let me clear this very nicely. Transfusion mismatch reaction is what? Suppose this is my blood group is A. So what I have? I have A antigen and B antibody. So this is the normal condition that is present inside my body. So if my blood group is A positive, so I have A antigen and B antibody in my A antigen on the RBC and B antibody in the plasma. So this is the normal things that will present in your blood group. If you give me a B positive group mistakenly, then what will happen? This will come inside my blood. You have given 1 pint or around 400 or 450 ml of blood inside my body that has A antigen and B antibody. Now you are giving me mistakenly. This is not obviously with influence, or we say with intention. It was, you don't know my blood group, or some reason you have given me another mismatched blood group. What will happen? This B group has B antigen and A antibody. So what will happen? This B antigen and A antibody. So you have given this to my blood, which I have a 6 liter of blood. So what will happen? This B antigen will be damaged, antigen antibody reaction will occur, and this all RBC that is you have transfusion to me will be damaged. They will activate this complement pathway, the classical complement pathway. Since there is antigen as well as antibody, this antigen antibody will be react, classical pathway will activate, and all this RBC will be lysed. All the RBC will be break down by the membrane attack complex. So all the RBC which you have transfused to me will be destroyed, and the byproduct will release in the environment. The complement pathway C3A, C5A that has anaphylatoxin and chemotaxis, that will cause vasodilation and people will go in shock. So due to hemorrhage, due to anything, you are giving blood to a person trying to save the patient, but if you are giving mismatched blood transfusion, then you are actually not helping that patient, you are actually killing that patient. So you have to understand this. So all the RBC which we have given, which is a B antigen, actually I already have a B antibody, and this all RBC will be killed, and all the byproduct will cause anaphylatoxin and even vasodilation and shock. This A antibody will also be there in this blood group B, and we already have A antigen. Even this A antibody will damage some of, because there is only 450 ml blood, so they will have less amount of this antibody obviously, but this less amount of body, whatever amount it is, it will come inside your blood, it will attack my RBC and destroy that as well. So look, if you are giving mismatched blood transfusion, there is actually a big devastating problem. The blood which you have given become useless, because that has all been bombarded, all has been destroyed in my body, okay, and there is a vasodilation as well, there is a shock as well, and the B blood group that has the plasma which has caused this A antibody, that will actually my RBC which has A antigen will also be destroyed. So this mismatched blood transfusion is a very severe disease, serious condition. You have to be careful. You should know the mechanism. Okay, so this is transfusion mismatch, activation of the complement, generation of large amount of anaphylatoxin and membrane attack complex that cause the red cell hemolysis, the given you have, and also some of the RBC of mine, and I will develop this hemolytic reaction, this acute hemolytic reaction, and you need to stop that blood transfusion immediately. Okay, so hope you have understood the complement pathway and its role, how this mismatch reaction, the complement protein, the complement pathway. If there is another blood group given to a person, then there will be a development of this acute hemolytic reaction, or say blood transfusion reaction. Okay, so there are some certain drugs that can be actually given to prevent this activation of the complement, like TNT 003. Then there will be a complastin CP40 and isolimumab. So these are the certain places where we can